Thanks, Dana. I should have probably mentioned this at the beginning. The session is going to be broken kind of into two halves, and we're focusing on the neuroimaging side with the first two speakers, then we'll take a break, and we'll have two speakers focusing more on the use of questionnaires and behavioral techniques to measure reward system responses and so on. So the second presentation in this part is by Hisham Ziauddeen. Did I get close? Good. Who is a psychiatrist working in the health science, neuroscience program and Department of Psychiatry at the University of Cambridge. His research focuses on the role of the brain reward system in normal and abnormal eating behavior using neurobehavioral, functional neuroimaging, and experimental medicinal approaches. He's currently working on mechanisms of antipsychotic drug-induced weight gain. And I'll turn it over to you. Thanks.
Okay, thank you very much for the invitation to speak. This is the title of the session. You will realize shortly that I have really seized upon the option of perspective that the organizers gave me, and they may regret that as we go through this talk. Before I start, I'd just like to make one acknowledgement particularly, and that's of Professor Paul Fletcher, who I work with, and the ideas that I'm going to take you through are largely our joint work. I, of course, will take full credit for anything that strikes you as completely outrageous or erroneous. Okay, the second thing I'd just like to acknowledge is my discomfort, which is the fact that I'm going to be critiquing various aspects of the published literature, and that includes the works of colleagues who are in the audience or listening on the webcast. And I just want to acknowledge my discomfort at that. I know as scientists we're used to critiquing the science, but I think as humans it's still not very nice when somebody critiques your work. Now, let me just try and figure this out. Okay, this is the talk. I'm going to be a lot more focused on the brief given and you'll see what exactly I mean by that. I'm going to speak specifically about neuroimaging techniques in the context of food addiction. So that's the context. This is the question, what can imaging technologies tell us about food behaviors? And this is the abstract of the talk. Okay. For about the first 15 minutes, I'm just going to be talking a little bit about the conceptual thinking behind this. And somewhere around the 15-minute mark, you'll be relieved when some data will appear, and then we'll wrap up 10 minutes from there. Okay, now when we start this particular discussion, in most cases, there's often the context of obesity, which is very much at the forefront of this thinking, and I doubt we'd be thinking about food addiction if we didn't have an obesity problem on our hands. I'm going to skip over that because I think we're all very aware of it, but I'd like to instead just look at this particular context. This I take very little credit for, apart from having put this in this kind of figure. This is actually the work from the Foresight report, which just looked at the various factors involved in mediating energy balance, which is essentially what lies at the heart of body weight. And I just put this here to emphasize the fact that, of course, we're all aware of, and that we've discussed at several points already today, about the complexity of the various factors that go into our understanding of intake and energy balance. And given this complexity, my point is, what questions can we meaningfully ask with neuroimaging when our overall causative model is so complex? And my take on that is that we can ask some very specific questions. And I want to just lay this out before I go into anything further. And this is the key as elements and assumptions that I think underlie most cognitive neuroscience experiments, and that's the region that we're talking about here. So we start with the assumption that we've got a phenotype that we are interested in, that there is a process that is meaningfully associated with that phenotype, or reliably associated with that phenotype that we're interested in imaging. We have a task that actually can examine that process, and our method allows us to reasonably examine the output of the task. There is, of course, two levels of control that are inherent in this design. One is, of course, if you're contrasting your phenotype with a control phenotype, but also the tasks involved inevitably do include a controlled condition. And that's because we know that we're not imaging or studying single-level processes. They're often integrated processes, so we have a a test condition in which we have the process of interest that we manipulate, and a control condition that we expect will control for all the other processes that are engaged by doing that kind of task. And I think my focus is going to be here on phenotype and process, but I want to just briefly highlight the importance of task, and I think that's very relevant when we're considering complex stimuli like food. And what I want to show you is, this is the only bit of my data I'm going to show you, and this is a task that we designed looking at a very common examination in this field, which is looking at high-fat versus low-fat food or high-fat versus non-food. This is specifically a motivational task. We're getting people to squeeze to win food. They're hungry people who are in the scanner who are essentially playing for lunch. And what I want to show you here is this contrast of high-fat versus low-fat versus non-food in terms of how hard people will squeeze for them. That's just plotted here in the force curve as opposed to a summary. And you can see that actually that particular task does produce some very robust neural activation when you look at the contrast of high fat versus non-food or just all food versus non-food. One of the things that's been troubling us in our lab for a while is actually how representative these things we do are. So when you show people images of food in the scanner, how close does that get to what you're actually doing outside? And how many assumptions are inherent in doing that? So what we did in a more recent task is actually we tried to control things a little better. We are often operating on this high-fat versus low-fat dichotomy. We used an objective energy density criteria of less than or more than 250 calories per 100 grams to define our high fat and low fat. We got a pilot group to rate all the pictures in terms of how much people liked them, how appetizing the foods were, and also how healthy or unhealthy they thought they were. And then we got participants to do the task and rate again how appetizing they found those foods and how much they liked them. And once you do that, your high-fat versus low-fat effects all disappear. So provided you control for the fact that all the pictures or all the foods are equally appetizing, they're equally edible, a lot of contrasts have been done where you're contrasting a burger with a whole cabbage, which apart from its appetizing or versus non-appetizing, also has edible versus not directly edible element in it. But once you control for that, we find that actually all our high-fat, low-fat, all these effects disappear. There isn't very much of an effect of healthiness or unhealthiness either. The last two columns here are non-fat and non-food and a positive non-food control, which is some rewarding stimulus. And if you break that up by lean, overweight, or obese, there are still no particular effects. Now, this is very early. I haven't got the imaging data to back this up, but having looked at this behavioral data, I'm not expecting the imaging data to jump out and bite me. So that's just something to say about the task, and I think that's worth thinking about. And this is the whole picture to be considered when we're evaluating what neuroimaging can tell us. And of course, this slide completely leaves out the most memorable aspect of these experiments, which is the brain images. So I want to just bear that in mind while considering two related, but I think slightly separate questions. Is there a shared similarity between drug addiction and a putative food addiction? And is there a shared similarity between drug addiction and food addiction literatures? So if we start with considering the phenotype, I'd start from this point of what do we think food addiction is? And I think there are at least two views, and they probably are more amalgamated as we've got along. Certain foods are addictive and they activate brain reward systems like drugs do, or certain people with obesity show a pattern of overeating that resembles drug addiction or drug dependence, and binge eating disorder is the most commonly considered candidate phenotype here. I put with obesity in brackets because I think it's a reasonable assumption that if we think there is a food addiction, there must be some people who have it who aren't obese, or at least are not yet obese. And if we just draw that parallel with drug addiction, certain substances are addictive and activate brain reward systems. In the drug addiction field, the thinking is that they activate brain reward systems like foods do. And certain people show a pattern of overconsumption of drugs that's described as drug addiction or dependence. But the reality is, of course, it's a combination of a drug and a susceptible individual that results in an addictive syndrome. And that happens in about 15% of drug users. And we aren't thinking very much about this at the moment, at least in the food field, but clearly if you are thinking of an addiction syndrome, there has got to be that element of time and an evolution of a syndrome in it. Bearing that in mind, here are 3 phenotypes that I think we have looked at so far in the literature. The first is obesity, the second is eating disorders, and this has mainly been binge eating disorder and bulimia on occasion, and then more recently, the syndrome of food addiction. Now, a lot of the work has been obesity. We talked this morning about lean versus obese comparisons and so on. I think obesity, as we all know, is an extremely heterogeneous phenotype. But a lot of the work that has been done is essentially use BMI as our measure of this phenotype. Now, Dr. O'Brien's going to tell you something far more useful and far more informed about the DSM and diagnostic criteria, but I want to just flag this up because the DSM-4 is what we've taken as our model for what food addiction might be. And I think, as a psychiatrist, I'm slightly skeptical of the DSM and the ICD-10 because I think you have to acknowledge them for what they are. They are sort of current best practice, best consensus guidelines. And of course, they have a lot of history behind them. And this graphic is just meant to sort of illustrate that actually, to get to this book, there's a lot that goes into it. And that needs to be borne in mind when you interpret the book, because you then can't just take the book and say, now that it is written, it is a standard, and therefore we will adapt this, taking this to be some sort of gold standard. The other thing to bear in mind from those DSM criteria is that they are criteria for a behavioral syndrome, and the criteria assume that there is a substance that's integral to the syndrome, and it's not designed for a substance like food that you need to take. The substance is, of course, another slightly tricky issue because what exactly is the substance? These are the potential candidates: high fat, high sugar, the combination, refined versus processed. And I think it may be any of these, but they're all terribly imprecise. And this is a pure hypothetical, thinking of addictive potential of a pizza versus the weight of cheese on it, and the relationship may be that way or that way. But I think without that kind of quantification, it becomes very difficult to actually know what you're studying. These are the DSM-4 criteria for substance dependence and their proposed food addiction equivalence. I'm not going to go through this in detail because I think that's going to be covered in the next session. I think there are a few things to note about these criteria, and one is that they do tend to conflate what we think of as hallmarks of the addiction syndrome with elements like tolerance and withdrawal, which very much relate to the substance you're taking. And there's a map-on of food addiction criteria from there. And I think those work as a reasonable equivalence, but there are some problematic elements in that don't actually map very well to food. I highlight tolerance and withdrawal over here, not because I think they're necessarily a big deal of our defining addiction, but I think if you're going to say a syndrome has tolerance and withdrawal, you need to have a good idea of what exactly that means. Okay. Ashley will probably talk about this in more detail, but the Yale Food Addiction Scale I'm just going to briefly touch upon because that's essentially our current tool for defining our phenotype of food addiction, and it's quite widely accepted. It is a sort of straight translation of the DSM-4 criteria from alcohol to food. It has had to make certain adjustments to the fact that you need to take food as opposed to not necessarily needing to take alcohol or cocaine, and has introduced severity criteria to demarcate what's normal from what extends into the bounds of pathology. And I think from a psychometric point of view, I do have some reservations about the Yale Food Addiction Scale, because one is, of course, relates to the compromise it's had to make in making the adjustment, but also in terms of the sample it was developed and validated in. And I think it was actually developed and validated in a young, largely non-obese sample. And most particular for me is the fact that there's actually a very strong correlation with a fairly straightforward measure of eating disorders. So what we're capturing is a measure that overlaps quite strongly with quite a Bando measure of eating disorders. It has subsequently been validated in other samples, and I think you can see there's quite a large overlap between the food addiction phenotype that the Yale Food Addiction Scale captures and measures like binge eating disorder. And also, this is just to highlight my concern again about the problem with tolerance and withdrawal. You can see that people endorse fairly high rates of tolerance and withdrawal in these studies. And that slightly worries me because I'm not quite sure what tolerance and withdrawal are with regards to food. Okay, so I think we have some idea of phenotypes, and there are certain limitations to the phenotypes we need to acknowledge before we consider the processes. And when we consider the processes here, we're largely guided by drug addiction. And this is just sort of a summary of the prevailing ideas of drug addiction. And there are about 6 points I've raised here. They're not entirely distinct, they're overlapping, and they're not entirely compatible with each other in all respects. But very broadly and briefly, the idea is that drug taking starts off as being voluntary and goal-directed and becomes habitual and compulsive. The drug taking sensitizes the dopaminergic systems and therefore makes drug-related cues more salient and motivating. And part of the idea is that as this goes on, the cues tend to be more rewarding than the actual receipt of the reward that they're related to. These are associated with impairments in the control systems that regulate behavior and also accompanied by a decrease in the D2 receptor levels in the striatum, and increase that as you continue with drug taking, drug seeking tends to become more driven by the need to avoid withdrawal and the discomfort it brings, rather than seeking the thrill of taking the drug itself. Again, as I said, these ideas aren't necessarily watertight, and I think there are lots of questions you could raise about these that I wouldn't go into for reasons of time. But I've highlighted 3 of them in bold here, and I've highlighted them mainly because I think these are 3 things that we've looked at in the food imaging field that are relevant. One, do food-related cues become more salient and motivating? Is there an enhancement of the anticipation that's triggered by a cue that predicts food compared to the actual receipt of the food? And is there a change in the D2 receptor levels? I think there are a few contextual issues to consider here. One is, of course, this idea that food and drugs act on the same reward systems. We should bear in mind that a lot of our understanding of drug addiction comes from the animal neuroscience of drug addiction, and not necessarily all these ideas have been fully tested out in human addiction. And there is a sort of risk of borrowed legitimacy, that because you find something with food that you happen to find in drugs, does that necessarily mean that your finding in food is addictive? And just to particularly highlight this point, I just like showing this data from Regina Corelli's lab, where she does detailed single neuron recordings from neurons in the accumbens, showing that actually you have very distinct neuronal populations in the accumbens that respond to water compared to those that respond to cocaine. And that isn't a feature of exposure to cocaine. It actually happens very early. So there are separate populations. They do occur in a very tiny structure of the nucleus accumbens, which we can't really image at that degree of resolution with human MRI. So you will probably find responses in the same region. That doesn't mean to say that they're actually happening in the same circuits within the accumbens. So those are some key contextual issues. More worrying for me are these key conceptual issues, and which are, is there an addictive aspect to normal eating behavior? And is there a fundamental addictive mechanism in the brain that controls normal eating? And the reason I pose these questions, I think, if you do a study in which you examine a process in lean individuals, or perhaps you contrast lean and obese individuals, and you say that that process means that there's something addictive, the assumption in that is that this is a fundamentally addictive process that we've imaged in a group. And I don't think that necessarily holds. I guess the simpler way to look at the question is there a neural signature of addiction in the brain? And I don't think there is a neural signature of addiction in the brain just yet. We have some clues, but there's nothing quite so definitive to say that if you find this, that means the process is addictive. So I've got into the data a little later than I promised you, but we are here now. These are the D2 receptor studies in obesity, and I think this is really what got the field moving way back in the early noughties. This is a seminal paper from Nora Volkov's group, led by Jean-Jacques Wang. And this is the first study that actually showed that there were reduced D2 receptors in the striatum in obese individuals. And it's an extremely powerful and compelling graphic. The graph below actually shows you those two groups visualized in terms of their BMI versus their D2 binding potential. And you can see there's a very clear separation between those two groups. I'm just going to flip that graph around and just take out the lines. And what I want to just highlight is that there is a fair bit of overlap in this D2 receptor binding. So you can see there's one person here who's got a BMI of 50 and one person here with a BMI of about 27, and their D2 receptor levels are fairly comparable. I just highlight the overlap there, not to say that that isn't important. But the means of the groups are clearly different, and that's really where the striking finding is. But this is just to put the distinction between the groups. All the people imaged here in the obese group have a BMI greater than 40. So we're talking about the morbidly obese. If we just flip that line to where we'd normally put our criteria for lean versus obese, so that's 25, and, oh, sorry. That's 25. I think there's still a stepwise pattern that you can make out, but I think that needs to be borne in mind when you look at this data. However, this is an extremely powerful study in the field. It's been replicated once. It's a slightly different study design, but close enough. But actually, thus far, there have been at least 4, if not 5, failures to replicate, and most striking of which is this one, which is also from Nora Volkov's group, where we have exactly the opposite effect. And while the first graphic that I showed you is the one that's most frequently cited, I'm not quite sure whether that really is a reproduced or reliable effect. Is there really a D2 receptor abnormality in obesity? There probably is one in morbid obesity, but if you're trying to look at more common levels of obesity, I don't think that's definite yet. Okay. Functional neuroimaging, here we're looking at 2 processes. This is our easy-to-follow graphic from our paper a couple of years ago. But I normally just flash this up and say, well, it's easy to follow, and there are arrows that go in all directions. But I'll just walk you through this. What we've essentially got here at the top are, these are the phenotypes we looked at from the literature. So that's obesity, and that's where you're looking at people comparing lean versus obese populations. Binge eating disorder, looking at BMI as a continuous variable as opposed to a dichotomous lean versus obese, and studies in food addiction. So those are 4 phenotypes, and these are responses to the presentation of food, which is, I guess, the closest proxy to food-related cues. This is a response to the anticipation of food, and these are responses to the consumption of food. And the anticipation and consumption are a particular paradigm that's been used a lot in the imaging field, which is essentially where you're getting liquid rewards in the scanner, and a cue tells you what reward is coming, but it's not a 100% predictive cue. Now that I've taken you to the graph, I will then tell you that actually there are arrows going upwards, downwards, sideways, in all sorts of ways. And I don't think there are any very consistent findings that emerge from this that tell us that there is one particular mechanism or any particular mechanism that stands out as consistent. There is one study that has specifically looked at the food addiction phenotype, and I'll take you through this in a bit of detail. The thing, again, I want to point out is similar to what I've laid out in the beginning. The phenotype here is food addiction. However, if you look at this sample in a bit more detail, let's just go there. So in this sample, there are, I think, 2 people who actually qualify for food addiction as per the criteria set out in the scale. So there are a lot of people who have food addiction symptoms, 2 people who actually have food addiction. And I think if you want to interpret these data, there are a couple of assumptions that you have to accept. And the first is that you accept that the Yale Food Addiction Scale is a valid measure of food addiction, and those symptoms do have value as a continuum of severity. So having 2 symptoms is worse than having 1, having 3 is worse than having 2, and so on. You have to consider the fact that in this sample, you're looking at people who have symptoms, not really people who have a diagnosis, even by the measure that's used. And even with that, what exactly did the results say? And this is the liquid reward in the scanner paradigm, where you're getting chocolate milkshake versus a neutral tasteless solution. And the prediction from the drug addiction field would be that you would get an enhanced anticipatory response to the cues that predict chocolate milkshake, and a decreased consummatory response to the actual receipt of milkshake. The relevant panels are these ones. So here, if you look, the black is a chocolate milkshake, the blue is the neutral solution. And there isn't any difference in the anticipatory response to chocolate milkshake, where the difference is in the anticipatory response to the neutral cue. And if you look at the consummation, this, by the way, on this left-hand panel is the high food addictions group. Again, you have a decrease in the consummatory response to both. I think that's just something you need to bear in mind when interpreting those results, but I don't think that that necessarily tells us that here's an addictive mechanism and here's a phenotype that clearly has it. I've often wanted to put this particular graphic up, and this is just my fantasy of if we were to imagine afresh how exactly we might go about looking at food addiction. I would think this would be how we might do it. Define an addictive agent, a syndrome that relates to it, find some way of measuring it, and then examine its neurobiology and its natural history. So far, we're sort of in that realm. And of course, if we really want to get that, we'd like to get some nice long-term prospective studies. Of course, what we actually get are cross-sectional studies that give us various pointers and similarities. A lot of this may merely reflect the fact that the field is in its infancy. But at the moment, to conclude, I think that actually there's very little direct neuroimaging evidence to support the idea of a food addiction. And there is a little indirect evidence, and we ought to be cautious in interpreting it, particularly given, from my point of view, the implications of actually accepting this model. And I think, for me, the overriding question in terms of how we might approach this is, what is the purpose of this investigation? Do we want an investigation of food addiction for diagnosis and treatment, or for policy change and improving legislation, for tackling the obesity crisis, or really as a scientific construct for further research? And that's important to bear in mind because we have different kinds and different standards of evidence and very different implications for each of those. And even if we're going to think about things like clinical diagnosis and policy, which are quite important and close to most of our hearts, there are still several tricky questions, even in terms of a clinical diagnosis. Who is going to be served and how would you serve them? And similarly, when it comes to policy, if you do come to the point of defining either an addictive food or a group of people who suffer from a food addiction, what are you going to do? And I think we could think of some things, but there are several questions that will follow, and it's by no means a straightforward conclusion. So thank you very much.